as an MSc project, to really think how you're going to organize your work, and to critically think the set of tools, because you can go into some of tooling wilderness, and I've done that. I've been there before, where there are just too many options, and everyone will always you'll always have a new update. Data will always change. Experiments will yield maybe good data, bad data, and that may make you may have to make decisions on what tool you have to use. So what I wanted to hear is more of the principles of how you set up a molecular biology, or rather a bioinformatics project, and we've all touched around file and directory organization, and it's easy to say, but you got to critically think. We've all said we're going to create top-level directory, we're going to arrange things, and that's technical. You're talking about your understanding of a basic Linux commands. That would be your utility. You need to know, how do I create a directory in a Unix? And partly you are all selected because you have that background, so I don't have to go into the details about Linux file structure. I'm going to assume that you know how that is structured. And most of the bioinformatics happen within Linux, Unix environments, largely Linux. So how you create your file, all the commands, and how you move files and directories from each other, you sort of, I would expect that, those are basics you already know about them, okay? But it just shows that that's really important. I didn't hear anyone talk about the lab notebook and how you keep that together with your project. Yeah, did you? Yeah. All right, okay, that group did. I need to get checked in. Yeah, so I was keen, listening in, how do you keep it? How do you organize your work? What are the principles of keeping a good lab notebook? And a lot of us talked about this, whether you're thinking about a single experiment, or a single project, and what results. And when you're doing that, you always have to make these decisions about what you're running, so how do you organize your work such that, you capture command line arguments? When do you use scripts, and how do you manage them? And when do you use programs? Okay. For example, I just got a new file of FASTA files, and the first instance I want to do is to ask the question, how many are they? Because that is going to inform me, or how large are these files, is going to inform me on my downstream processing, right? I don't need to write a script to do that. I will just run a set of command line options, grep minus v, grep minus c, quotes, that sign for whatever. Can I just do this? Just quickly. Yeah, so if I want to know, I can do this. Okay, and that will tell me, you simply count how many files I have in my script, or in my input file, right? If they're in tens of thousands, that's also important, right? So if I'm starting my project, how do I track that? Okay, so some people, what they do is that they always pipe their command line. So you'd start your directory, assuming that what we're doing here, make dir my project, right? Cd my project. That's what we're talking about. So we're in my project. It's an empty directory, isn't it? So the first thing I'll do, I'll organize it in the way we all did. I'll create a couple of directories. Make dir minus p data, right? SRS for source, maybe figures for when I'm going to keep my figures, supplements for where I keep my supplements, or supplementary data. And if I do that, I have the directory structure, right? And one of the things that we all mentioned, and I think one of the groups said, was that, yeah, there's one more directory here we want to create, which is the results. Okay, is that within the results directory, they will track them by time. Okay, so they will create subdirectories where the day's input is stored in that file. What's the flow with that? What's the problem with doing that? It was your team. So you say that, was it? Yeah, that one way to keep track of our results folder is that all the day's analysis would have subfolders where all that day's results are stored, right? And that means that for the duration of the 365 days you're working on that project, you have 365 directories. Isn't it? How can we control that or mitigate against that? Git init. We use source control. We don't need to keep 365 directories. I can simply use source control, right? And every day I'll run my analysis. I'll have my git ignore. I'll know which results to keep, which ones not to keep, and then I'll let git make keep track of those results. And the good thing with that, I don't also have to worry about naming the days, and I'll always have the same directory structure. Okay? And if I expand it, contract it, I have a complete history, and I'll know exactly when each result was stored. Okay, so that one way to do it and to think about it. Okay. So this is a nice paper, quite old, but still very relevant. That you got to keep about this. And I was saying that what some people do is that they pipe and they store everything in their directory. Unix does this by default. So there's something you might note. You can always go to your command line and look at history. So these are the commands that I've run today. I got a small script, the way I've set up my bash, to remove redundancies so that the same command, if it's typed twice, can be cleaned up at the end of the day so that I can keep my history. But I got a lot of history here over time across all the commands that we've run. Okay? And I can always go back to that history and retrieve it. So again, the history command, and if you do, I think, if you're looking even for a command, I can always do this, and I can pull it from history. I can pipe that request and retrieve it from history. So for example, if I run just history here, and I pipe the output of it, and I'm interested to see all the Docker commands that I've run for the day, all that, and if there are too many, I can pipe that to another command that now allows me to tab through each of these, over time. And the number you see at the start here, if I want to rerun it, I can simply rerun using that number. Just ask, can you rerun that command again? But the point is that I can capture that and I can pipe whatever I done within a single directory into such a file. So I now have a complete history and of all the command line options and arguments that I've run, and I can keep that in a separate file. Okay? And the principle here, which I like, and you see it in this paper, is the idea that, I don't know whether I can blow this here. I like this, it's quite difficult, that someone who is unfamiliar with your project should be able to look at your computer files and understand in details what you did and why. And that person will be you in 3 months' time. Often. Okay? Yeah. Particularly if you're doing your PhD, MSc, or working multiple projects, that person will just be you. Can't remember what you did, you struggling to remember what code you ran, that smart move that you did, and you can't remember it anymore. Okay? And the same thing, so try and document your stuff. And scripts, again, can live in a script folder, and the programs here are, tools that need to be installed. They could be the BWA, could be a specific tool, and also the idea of environments and virtuals come into play. And version control is really useful, as I just showed. Backups, tracking down bugs if you're writing your code, and it's invaluable for collaboration, okay, and working together. So this again is a diagram from the same paper, showing pretty much what we've talked about. Sometimes what I tend to do is that I'll have a huge directory. All the programs, I'm going to put them in the bin directory, right? I'll put them in there. I'll set it up such that I'm running from those directories or from the bin. Source is where my scripts will go. Data is where my data goes. Documentation, this is where you documented things, and from this, your paper is going to be generated from that. And then again, you got results. These are different days. I'll just use Git to manage my results folder. And data, you can have multiple data sets, and you can subset those by dates if you want, or find a different way. Again, for most bioinformatics, computational biology projects, it's not just about data. It's not just about this organization. You've got to think about data governance, right? If you've been working on SARS-CoV-2 or anything, you know that you need to protect your subjects, and that not everything needs to be visible sometimes to everyone. There's sensitivities around it. So whenever you're thinking about your data analysis, your project management is actually about data. So I borrowed a couple of slides here from Farid Arikan. He's a great bioinformatician from Quadram Institute. And this is a course he gave, the Kleibo workshop. And I just love this diagram here, where you're spending time here gathering. You got your experiments here. You're generating your sequence data. You also have published data streaming into your analysis, and this is sort of a contextual. So for example, you may generate new data, but then you need to compare with the data that's already existing, all from your previous experiment. And over here, you're running scripts, you're doing analysis, you're integrating data here. And this is sort of your work area, and they are all, visualization. It's sort of feedback. Whatever you do here, you feedback the analysis and so on and so forth. And you got to think about now, how do I share the results of my great work? That could be either publishing, sharing with collaborators, open access. What aspect of it do you make open access? And how do you securely transfer this data to your collaborators between organizations? And you got to think about archiving and storage. Okay, so the lifetime of your project, maybe it could be an MSc thesis or a PhD, and at that point is the greatest thing, and after a while, it's no longer, it's outdated. And it's still important, so we need to think about how we're going to keep all this and archive it, and think about policies before either destruction, maybe we need to recover space because you only sequenced 10 nucleotide gene, and the technology now is allowing you to do everything. Okay, so it's been surpassed. Whatever, yeah. So you got to think about that. And what Farid does is again show this this plot here. You know that our results should be reproducible from primary data. Okay, so here you feeding in your primary data, contextual data. You got to think about the workflow and software, your analysis software here. You know, and you got all these secondary data, your raw results. Tertiary data is what is coming out from your primary, secondary data. You know, this could be figures, this could be tables, manuscripts, you know, things that you want to share. And around here, you're thinking about visualization, tool scripts, and how you're going to communicate those results. Okay? And therefore, the things we are learning, you've got to think about where do they fit in and how do they, you know, become part of this workflow. Okay? So for workflow and software, you know, you can think about SnakeMake Nextflow. We're going to cover SnakeMake and see how we can, you know, automate scripts. And, you know, some of those command line things can be taken care of by storing them nicely, reproducibly through SnakeMake or Nextflow. But you got to pick one often. Okay? You can work with either, but, you know, that's can be subject to, you know, either what you're introduced to first, but also what works best in your environment. Analysis software here, you've got to think about Coda, Docker, Singularity. Again, important, you're going to choose one of them. It's always nice. And, you know, you can think about project-wise, where I will say this is a project I'm doing. I'm going to, you know, the features or the requirements for this project. You know, if you think in big term, you know, rather than think at the moment, it's always nice to take a step back and say, okay, so these are the requirements for the project. This is the durability of the project. What am I going to use? Is it too much of an overhead to write SnakeMake scripts, whereas I could just do a quick 3-liner bash script? If it got lots of moving parts and so on, I might want to script it. If I want to share it with collaborators, I want to script it. So then I'm going to use SnakeMake or Nextflow. And you know, is it all Python packaging and whatever? Is it something that I can just use Coda, easy peasy, run a Coda environment, containerize that within that environment? Or am I going to use Docker? Do I need to pull things other people have already used? Has somebody already done what my PI has asked me to do? And there's something already there. There's nothing as good as, you know, being asked, can you do X, Y, Z, and tomorrow you got the results. PI often doesn't care, honestly, how you did it. Okay? It's great if you're doing it, but results or this one over here. Okay? As long as it's reproducible, as long as, you know, you can document what exactly you do and you know what are the moving parts. Okay? You're a bioinformatician, you're an engineer, you need to know and repair your engineering work. Okay? So again, I really like that diagram there, and it summarizes pretty much what we're doing today, or rather what we've done, and I hope now you can contextualize what we've been doing the last 2 days, yesterday and today, and then tomorrow we're going to come in here and see how best we can create these workflows and take advantage of the analysis software that we've already now familiar with or have set up to move this work forward. Okay? And with that, any questions, discussions? Yeah, it's 9 minutes to 4. Yeah, this is available. Yeah, this is available from the clip. I'll put the notes in. It's really nice, and it's quite a nice talk to listen to. Farid Khan giving that talk. It's on YouTube as well. You can, you know, I really highly recommend. Yeah, but I hope it all fits together now. Okay. Yeah. Awesome. Yeah.